Another day, another story. Immunological techniques are laboratory methods used to study the immune system and its responses to different antigens or pathogens. These techniques are used to identify, quantify, and characterize immune cells, antibodies, cytokines, and other immune-related molecules. Some of the commonly used immunological techniques are 1. Flow cytometry, a technique used to analyze and sort individual cells based on their physical and chemical properties, such as size, shape, and surface markers. 2. Enzyme-linked immunosorbent assay, ELISA, a technique used to detect and quantify specific antigens or antibodies in a sample, based on binding an enzyme-labeled antibody or antigen to the target molecule. 3. Western blotting, a technique used to identify and characterize specific proteins in a sample, based on their size and immunoreactivity to specific antibodies. Polymerase chain reaction, PCR, a technique used to amplify specific DNA or RNA sequences in a sample, which can be used for various applications, such as gene expression analysis, genotyping, and sequencing. These techniques are essential for understanding the immune system and developing new diagnostic and therapeutic approaches for immune-related diseases. Here are some brief explanations of the 4 techniques mentioned. Flow cytometry. This technique is used to analyze cells and their components, including immune cells, by suspending them in a fluid and passing them through a laser beam. The laser light is scattered by the cells, and detectors measure the amount of light that is scattered at different angles and wavelengths. This information is used to identify and quantify different cell types and their properties, such as size, shape, and surface markers. ELISA, enzyme-linked immunosorbent assay. This is a commonly used laboratory test to detect and measure antibodies or antigens in a sample, such as blood or urine. The sample is added to a well containing a specific antigen or antibody, which binds to the target molecule. A second antibody linked to an enzyme is added, which binds to the first antibody or antigen. A substrate is then added that reacts with the enzyme to produce a detectable signal, which can be quantified to determine the concentration of the target molecule. Western blotting. This technique is used to detect and identify specific proteins in a sample, such as a cell lysate or tissue extract. The sample is separated by electrophoresis, which separates proteins based on their size and charge. The proteins are then transferred to a membrane and probed with a specific antibody that binds to the target protein. The antibody is then detected using a secondary antibody linked to an enzyme or fluorescent tag, which produces a signal that can be visualized and quantified. PCR, polymerase chain reaction. This technique is used to amplify specific DNA sequences in a sample, such as a blood or tissue sample. The DNA is first denatured by heating, then specific primers are added that anneal to the target DNA sequence. DNA polymerase is then added to extend the primers and synthesize new DNA strands, which amplify the target sequence. The amplified DNA can be detected and quantified by gel electrophoresis or other methods. PCR is commonly used to diagnose infectious diseases and genetic disorders, and to study gene expression and regulation. Thanks for watching.